Hi, this is the review video for Lecture 6, Microbial Genetics and Biofilms. And as you may have noticed going through it, it's not all genetics. Biofilms are distantly related to the topic of genetics, and the connection between them is something we no longer discuss in this class. So it is what it is. We start with the biofilms, and you have to know what they are and why cells make them. And you have to know that this is where we find bacteria in nature, generally. Where do you find them? You find them where there's water flowing over a solid surface, because bacteria want to be on that surface. They want to hold on to it and have nutrients come to them and their wastes flow away. But also, they build these things to protect them from predators. So they're not individual cells that can be eaten by protists. They are giant clumps of carbohydrates and protein blobs that cannot be eaten by something as small as a protist. So biofilms are everywhere where a solid surface meets water. So every rock in the ocean, every rock in a river, teeth, and they would be on the insides of all your blood vessels if your immune system didn't prevent that from happening. So that's biofilms. That's the main thing I want you to know. And you can visualize this is kind of the life cycle of a biofilm, so to speak. And one key thing to note is that the cells come in with flagella, because that's showing that they are optimized for living alone and moving around. But then the cells in the biofilm are drawn without flagella to show that they are now behaving in a way that makes more sense if they're in a biofilm. So they change their behaviors, such as not making flagellin proteins. And then once the biofilm is very mature, cells start having to really compete for nutrients, and some of them will intentionally leave and go back into their individual lifestyle. Okay, so that's biofilms. Then a bunch of stuff we skip, and then mutations and evolution. We don't really get into evolution, but we look at kind of the basic raw material of evolution, which is nucleic acids changing through mutations. So the first thing we look at is 5 different types of mutations, and what do we think about? We think about how a change to the nucleic acid sequence changes the amino acid sequence of a protein. So what effect does a mutation have on the protein that is encoded by that nucleic acid? And mutations, we do sometimes want to step back and think about what are they, and they are heritable changes to a nucleic acid sequence. That means they have to be in the nucleic acid that gets passed down to a daughter cell, or that gets copied to make a new virion. And we bring that up specifically because there are other nucleic acids in cells, so not in viruses, but in cells. There's DNA, and then there's RNA that's made by transcription. And RNA can have errors just as well as DNA can in a cell. So DNA replication is usually where mutations happen, but transcription is not perfect. But the thing is, the proteins made during transcription in a cell are not passed down to daughter cells, generally speaking. So they're not mutations, they are just errors. So that is a difference. And it gets tricky because viruses, if a virion is made with a nucleic acid that has an error in it, it will try to replicate that nucleic acid with the error in the next host cell it goes to. So that is heritable, whether that virus's genome is DNA or RNA. Okay, so whatever, DNA, RNA. So that's what a mutation is, and what you need to do is learn the 5 names of the types of mutations. So all mutations fall into one of those types. So make sure you know the types and what we call them and what effect they have on the host cell. And let's see, what else is in my study guide for lecture 6? Biofilms, a bunch of stuff crossed out, and mutations, and then error rates. So how often do mutations happen, and how would we state that? And this, again, is something you really have to think about. What are the numbers that I give you, like 10 to the negative 6, or 10 to the negative 4, or 10 to the negative 10th power? Like, what is that about? And I want you to remember that we're always thinking in terms of mutations per numbers of nucleotides. Okay, so a human or some eukaryotic cell might have something like one mutation every 10 to the 9th nucleotides. That is to say, when a cell from an animal or a plant is copying its DNA, it is doing a fantastic job of copying it perfectly. It makes one error out of every 1 billion nucleotides. That's like typing a book 1 billion letters long and only making one error. That's not something a human, like, intentional process could ever do, but all of our cells do it all the time. But we don't use this fraction in calculations. We don't write it this way. We write it as if we had gone ahead and done this division, because that's the same as 10 to the negative 9 mutations per nucleotide. And this number doesn't make sense if you think about it. Literally, you can't have a fraction of a mutation in a nucleotide, because that's really what this says, is 1/1 billionth of a mutation in a nucleotide. Well, that's not possible. What is possible is if you look at a huge number of nucleotides, this is the average number of mutations per nucleotide. So it's kind of like the chance of a nucleotide having a mutation is 1 in a billion. That's what this number means. So the bigger this number is, or by contrast, the smaller this number is, the more accurate that process of copying the nucleic acid is. So make sure you look at the slides that compare mutation rates from like the higher eukaryotes that have a really slow rate of mutations compared to an RNA virus that has a high rate of mutation. And which one has the bigger number? Well, the bigger number is here, 10 to the minus 5th, is much bigger than 10 to the minus 10th or 10 to the minus 11th. So make sure you get that. And there are two examples of why these error rates matter. One is the example of HIV, the human immunodeficiency virus that mutates so quickly we cannot make a vaccine against it that works. And a lot of what's mutating is going to be the spike proteins that are always in danger of our antibodies detecting them. And then the other end of the spectrum is smallpox, which is a DNA virus which mutated so slowly that one vaccine worked for several years, and we were able to completely eradicate that virus. And now it only exists, in our knowledge, it only exists in 2 government labs in the world. Realistically, you can't trust any government to not sell a weapon to another government. And you'll see in Unit 4, we'll learn bioterrorism agents, and I will refuse to call them that because terrorists don't use them, armies use them. So get ready for that. I have opinions. So what else? Keep going. And the next topic is horizontal gene transfer. And for this, here's what you need to know. You need to be able to tell me the 3 different types of horizontal gene transfer in bacteria and basic description of each of them. Because these are ways that one bacterial cell can give DNA to another bacterial cell. So not to its daughter cell, but to its neighbor. And this is how genes in bacteria move from species to species within a hospital. And you can document it happening in someone's gut, where a bacterium that has a gene that makes it resistant to an antibiotic can give that gene to another bacterium. So now you have two different antibiotic-resistant species in your gut. That only matters if one of them causes a disease and you try to treat it using that antibiotic. Otherwise, it doesn't matter. But it turns out in hospitals, lots of people have diseases we try to treat with antibiotics. So this is a terrible thing. So there's that. And then that brings us to antibiotic resistance, because this is, depending on who you ask, the biggest problem facing healthcare. And healthcare systems are completely unable to stop this. This is going to beat us. This is going to put us back into the era of most bacterial infections being deadly. Hopefully not in our lifetimes, but antibiotic resistance is your enemy. So you have to understand what exactly this means. And there are two lists that come from this that you have to memorize. One is, what are the mechanisms of antibiotic resistance? So what do cells do that allow them to be resistant to an antibiotic? And that's somewhere in here. Mechanisms, yeah, this list. These are three of them, and then figures that might help you envision them, and then two more. So the first four are the most important. The fifth one is kind of rare, but it was one of the first ones that was discovered. And then, how do we delay resistance? This is an even more important list, because this is something that clinicians can use to prevent the extinction of humanity. So please learn how to delay antibiotic resistance. What else? What else is in the study guide? That's it. So, right, that's it. Everything else here is curiosity slides for a topic that we don't have time to go into. Look at that cute squid. I probably know the person who took that picture, but I've never met one of these squid. They're very small. And, oh, right, this is great. This phrase, you will hear people say this is meaningless. So please read through this. What does it mean when someone says, I became resistant to that antibiotic? What that means is don't accept medical advice from that person, because they don't know how it works or what it means. So you should become the person who can say what they're trying to say in a meaningful way. And there you have it. That's lecture 6. So go forth, study, and I will see you at the next video.